Well, it's good to be back. I've been here all day visiting with labs, and I've really enjoyed it. So I appreciate you guys. And yeah, I'm just here today. I'm going to talk to you about my career path and just kind of tell you what I've been up to, what I plan on doing this year. Starting from just taking a general entomology course all the way to state entomologist where I am now. So yeah. This is just the outline of my talk. I'll briefly talk about myself, and then I'll go into my educational background, and then what I did after I got my degrees, so my early career, the jobs I did there. And then I'll, of course, talk about my current position, as well as kind of give you guys an update on what I plan to do this year. So my family and I are originally from North Carolina, and I moved to Kansas when I was very young, so I've been a Kansan most of my life. If you guys don't know, I have a dog. Her name is Pebbles. She's a Chihuahua Terrier. She's my pride and joy, and basically the best dog. So, and then yeah, when I'm not talking about insects and doing entomology work, some of my other hobbies include gardening, whether that be growing crops or flowers, and then also native plants. So I'm a huge advocate for planting native and landscaping with native plants. And those are just some of the pictures of some native plants that I have around the house. So with that, I'll jump into my educational background. So I was an undergraduate. I started here at Kansas State University in 2014. I knew right away I wanted to get a bachelor's in biology. I've always liked the outdoors and wildlife. I didn't necessarily know what I was going to do with it yet, but I knew that's what I wanted. And then a little later down the line, I would eventually get my entomology minor. During my undergraduate time in 2016, as a sophomore, I did my very first research project. It was at the first annual undergraduate research symposium. This was brought on by Dr. Jeremy Marshall. He basically came to our general entomology class and brought up the idea to see who would be interested in doing kind of like a mini research project. And at the time, I didn't have a lot of experience in research, but I really enjoyed learning about it. And so I took on a project for that fall. My supervisors were Dr. Chris Silver and Dr. Kunyan Zhu, so I worked with them for the first time. And my project was using RNAi against target genes of the German cockroach. So I would go in every so often and inject dsRNA into the abdomens of German cockroaches. And, I wasn't swayed from it, and I fell in love with research. Kind of at the same time, after I finished that project, I was kind of, I was minoring in entomology. So I was kind of more involved with the department, and I came across a job posting for a biological sciences aide or a student worker over at the USDA over on College Avenue. And I applied for that job, and I think during that time there was a government shutdown, so I was a little delayed in starting, but I got that position, started kind of in spring of 2017. My supervisor was Dr. Aaron Scully, but I was also kind of split up between the Morrison, Scully, and Campbell labs. And, this was kind of like my first job really utilizing my degree. And I learned a lot just from doing this kind of part-time work. Obviously, I learned about stored product insects and the whole world that is stored products, which I had no idea about at the time. I learned how to do just basic lab maintenance, how to, maintain insect colonies and just general upkeep of a research lab. And then, I, of course, learned important skills like data collection and following protocols. And then another big thing, another hurdle I had to, overcome being a student and not having too much experience was just accepting failure and knowing that you're going to make mistakes, especially in research. It's really common, but, keep working hard and keep pushing through. And so that was another big skill I obtained. And then, so I worked that position for a while, and I was nearing my graduation with my undergraduate degree. And I was asked, do I want to stick around and maybe do my master's in entomology? And so, at the time, I wasn't really sure what I was going to do with my bachelor's. I had enjoyed doing this work over at the USDA and being involved with research. And so I was like, yeah, I'll stick around. I'll get my master's. And so that's what I did. So stayed at Kansas State. I had two really great advisors, Dr. Rob Morrison, Dr. Kunyan Zhu. Since I did most of my research over at the USDA, I was primarily part of the Morrison lab. And Dr. Morrison couldn't have asked for a better advisor, and I learned a lot working in his lab. My research was about diversifying store product integrated pest management programs, or IPM programs, using long-lasting insecticide netting, or LLIN for short. And since I only have a limited amount of time today, I'll just briefly kind of go through what my graduate research was about. So I looked at the netting that was incorporated with an insecticide and looked to see how we could use it to prevent, adult species of store product insects from dispersing into food facilities and getting into commodities. So we looked at the netting's effect on adult movement and dispersal. We also looked at the netting's effect on immature stages of store product insects and how the netting affect their movement and dispersal. And then we also looked at how, different ways you can deploy the netting in these pilot scale warehouses, as well as incorporating the netting into other IPM tools like attract and kill. And, with my graduate research, this was, again, another big growing point for me. So I gained a lot of skills throughout my master's program. One of those being, presenting. That was a huge skill. I kind of overcame, my fear of public speaking. I still have a fear of public speaking, but I've gotten better at it. So just being involved with the entomological conferences and like ESA, KES, the Kansas Entomological Society, being involved with those, doing student competitions, that really helped me with my presentation skills. Also, just here at the university, we have the 3-minute thesis. I competed in that. My first year did pretty well, got to the finals. And so that was just a great way to, practice how to talk about your research to audiences that aren't very familiar with what you're doing. And of course, the K-Grad research forums was another outlet that helped me work on my communication and presentation skills. Also, I learned how to do, practice, or I gained skills in publications and writing. So just from all the work that I did during my master's program, I came out with 5 peer-reviewed papers. So I learned a lot about, collaboration and the publication process. Another skill I gained was leadership and teamwork. So apart from just taking classes and doing research, I was involved in the debate team for ESA. I was also involved with the entomology games trivia team, just so some other fun ways to improve my skills and maybe not think about research for a little bit. And then, of course, I was involved with the Pocono Club. I had a couple of officer positions with that. And so that kept me involved, and yeah. And then finally, outreach was another big component of my graduate program. And there were so many outlets for doing outreach, whether it's with my lab or just out in the community. And, outreach is so important, especially because, you're learning all this new information. And so, what are you going to do with it if not share it with other people, and that maybe they could benefit from learning all this new information. And so really, every job I've had, even today, outreach has been applicable to every job. So it's very important. And with that, I'll move into my early career. So what I did after I graduated with my master's. 2020 was a great year to graduate and look for a job. I moved to Springfield, Missouri, while also still continuing to look for a position, which was a little difficult, especially when you're looking for entomology positions in a town. Kind of hard to do, but it is what it is. I started looking at, state jobs. I looked at Missouri State University, which is there in Springfield, Missouri. And they had jobs available, but I found, there was a lot of internal hiring for those kinds of tech positions that I applied for. So oftentimes I would apply and just not hear anything. This university didn't have an entomology department, but I was able to reach out to a professor there who he worked with bees. And so I reached out to him, but he had limited funding, so I was not able to start right away. However, being in a new town, I decided to kind of stay active with the community, maybe work on some other skills of mine and some other interests that I had. And so I did community service, working outside. I went to the Springfield Botanical Gardens. And, if you ever find yourself in Springfield, Missouri, I definitely recommend going to these gardens. It's probably one of my favorite places. So I decided to sign up, do some community service. I adopted the Lake Drummond Garden, which you can kind of see in the picture, but it's a little hard to tell. It's across the lake. And I just spent some time working on, invasive plant removal among the garden. There's a lot of Johnson grass, Sericea lespedeza. These gardens were native plant gardens, so I helped establish native plants. And then just general garden maintenance, like mulching, weeding, things like that. And then, I was still looking for a position, and I came across one that kind of piqued my interest. It was a little different from my, master's research in entomology. It was called AmeriCorps Vista. And when I came across it, I didn't know what that was at the time. And if you don't know what AmeriCorps is, it's kind of like a federal service program that helps nonprofits and other organizations who maybe don't have the money to pay for, employees and staff. And you apply as an AmeriCorps, and they send you to these organizations to kind of serve there. And these organizations can be in a variety of different sectors. So like environmental stewardship, health, education, disaster relief. And for me, the site that they were advertising was for the Watershed Committee of the Ozarks. So that was an environmental group. And, I was really ready to get a job. This was environmental work, and that's definitely, one of my interests. And so I applied for this position and I got it. And so I spent a year working with them, and their mission was to improve water resources and drinking water through education, outreach, and watershed management. So my official title for this position was development coordinator. So I had a lot of different responsibilities. One of those was creating, outreach activities surrounding water quality. So for example, one way to determine water quality is to look at the types of benthic macroinvertebrates that live at the bottom of streams. And depending on the species that are present or absent, can kind of tell you the health of the stream, because some of those species can only tolerate so much pollution, if at all. And so I came up with different activities around that, which I really enjoyed. I also helped with field trips at the Watershed Center, which the Watershed Center was located in a park. So we had, students coming in. We had volunteers trying to get service hours. We had businesses come in for Day of Caring events, which was a national service event. So just leading those kinds of field trips and those kinds of groups. I was also trained as a water quality monitor. So the great thing about AmeriCorps was like, yeah, you're helping out this organization, but they also want you to kind of gain, skills that you can maybe use in your future jobs. So being trained as a water quality monitor through stream teams was kind of cool, and definitely, not entomology related, but, I really enjoyed it. And then finally, another thing I did was learn about invasive species and native plants, hence my interest in native plants. The organization I worked at had a native plant nursery, so I helped with, seed collection, native plant sales, planting seed, and even helping them build their greenhouse. And of course, having an entomology background, I wanted to put my own spin on this position. So what I did in my free time was I created an insect collection from insects found around the park to kind of show visitors that come to the park, like, hey, look at all these insects. If you take care of the land around you, you can have some really cool diversity. And so here's just some of the photos I took of some of the insects I found around the park. Just some really, really cool insects. And yeah, I had a really fun time building this collection for them. And then, I left the collection with them once I finished my time in AmeriCorps. Yeah. And During the same time, once I started my AmeriCorps position, the professor I had reached out to at Missouri State University, he reached out to me saying like, hey, I now have funding for a part-time research associate position. And with AmeriCorps, you're allowed to do part-time work since they don't pay you super well. It's more of a service-based type of program. So I heard from Dr. Avery Russell, his lab. He offered me a research associate position. I was kind of really excited to get back into research a little bit. So I readily, interviewed with him and got the position. And then I started working in a bee lab. So a little bit different from my store products lab. We worked with bombus impatiens, so the common eastern bumblebee. And although he was a bee lab, his research was in a lot of different things, behavioral ecology, and he did a lot of plant insect microbe interactions. So yeah, just some of the photos on the screen there. That's a bee that's on the camera when it should be sonicating flowers. And then on the right side is just some flowers. That's kind of like one of our setups that we would do for a behavioral trial. All right, and so with this position, again, I gained a lot of new skills. So learning how to count and identify different types of pollen. So on the left there, that is a lot of pollen under a microscope on a hemocytometer. And so I did a lot of pollen counting. I learned how to do acetylysis. If you don't know what that is, it's a multi-step chemical procedure that breaks down organic matter. So for our lab, breaking down plant material in order to count the pollen. And of course, I learned how to handle bees and not get stung, although we did have, an EpiPen handy. But yeah, bees were very different from store product insects. They had their good days and bad days, definitely a lot of personality. I think by the end of it, I was starting to miss my little red flower beetles. So, and then my supervisor that I worked with, he was very supportive and, wanted us to, do our own kind of research project if we were interested. So I also kind of independently led my own project. And for me, I looked at how anthropomorphology affects pollination success in flowers that are closely related to each other. So just to quickly kind of go over this project, it did get published. So if you want more information, feel free to check it out. But, the idea behind it was, there's these plants whose flowers, the anthers are very different in terms of their structure, but yet they're closely related. So, what are the pros, what are the cons to having these different anther structures? So for this research project, we had two treatments. We had flowers with joined anthers, which the anthers are naturally kind of fused together into a cone shape, like in the photo, letter A. We also we compared the joined anther structure to free-moving anthers. So obviously the anthers are not joined together, as in letter B. And so for this experiment, we had our study species, of course, our model pollinator, bombus impatiens, and then we had Solanum malagnifolium, the silverleaf nightshade. And for that Solanum malagnifolium, they naturally have free-moving anthers. So what we did for the joined treatment, we would just glue the anthers together. And so, yeah, we allowed the bees to pollinate the two different types of anther structures. And we took multiple different measurements. So we measured the pollen that was remaining on the bee body after sonication, which that pollen might be available for pollination and would be beneficial to the plant. We also measured the pollen that was received by the stigmas, the pollen remaining in the anthers. We also looked at the bee itself. So we looked at bee body size and how long the bees spent on the flowers. And just, quickly going through my results, we did see that there was a difference between the two different types of anther structures in terms of pollination success. However, this is dependent on the pollinator itself. So we saw that larger bees were better pollinators for free-moving anthers. And we kind of concluded this because we saw more pollen remaining on the bee body for pollination and more pollen that was deposited on the stigmas. We also saw that smaller bees were better pollinators for joined anthers. So we saw that less pollen was collected in their pollen baskets and more pollen was deposited on the body and the stigmas. All right, so while I was doing these 2 temporary positions, I also wanted to kind of show that, I am I'm learning these new kinds of skills, but I kind of wanted to get that put on paper. So a friend came up to me and suggested I get certified as a private pesticide applicator. This kind of work was really relevant with my AmeriCorps experience, as well as doing the community service. So I thought it would kind of bulk up my CV a little bit, maybe help me look for future jobs. And so it was a really easy process. I did it through the Missouri Department of Agriculture's pesticide program. And all it was you needed to attend an online training seminar. There was no license fee, you just had to purchase the manual. So that was one certification I was kind of excited to get, just to kind of show like, yeah, I have this background, in pesticide application and environmental work. And then at the same time, in the spring of 2022, I was doing some contract work. One of my colleagues came to me and was telling me about one of their research projects and how they needed to identify a bunch of different insects in these trap samples. And, I really enjoy doing taxonomy and systematics and identifying insects. So I told them like, hey, I can help you out. So I did a little bit of contract work, again, through Kansas State University, identifying different store product insects in my free time. So just some of the photos on the screen are just a few of the genera and families that I found. So I was, this kind of position or this kind of work kind of helped me bulk up my ID skills and, keep me learning even when I didn't necessarily have a full-time position yet. Which leads me to my current position. So later on that year, in 2022, I saw the job posting for a state entomologist. I applied for the position. Even though it was in Manhattan, Kansas, I knew this was a job, I could not pass up. It sounded too cool. And so I applied for the position, interviewed in June. And then I got the job and I started working in July. So that meant coming back to Manhattan, Kansas, and working with the Kansas Department of Agriculture in their plant protection and weed control program. So, I've been asked a lot, what do I do as state entomologist? And so there's a lot of different responsibilities that I have with this position. In general, I help develop and manage the entomology program for the state of Kansas. I also kind of stay up to date on the latest pests and any kind of incoming pests that we need to look out for. So I provide reports on those, do pest risk assessments of any kind of pest that might be, might potentially cause agricultural or natural resource concern. Also, we have a lot of area inspectors around the state. And so, when I have some time, I'll travel to different parts of the state and help them with their nursery and greenhouse inspections that they do every year. They do random inspections, but they also inspect any kind of greenhouse that does exports. So I'll go out with them and just walk through the greenhouses, looking for insects, which, couldn't ask for anything more. Looking for insects as a job, it's pretty great. And just any other kind of plant diseases that stand out. And then, if we see anything, just letting the managers know. Yeah, and then I do a lot of collaboration as well. So I haven't been working here a full year, but I've already been collaborating with USDA APHIS. In the top photo with the trees I took, that's just, we did a survey for ash trees in counties that recently discovered emerald ash borer. So looking for ash trees and looking for sites that could be potential release sites for EAB biocontrol agents. So I went out with a pest specialist for APHIS and we did that. Also, we work with the Kansas Forest Service. So last fall, I went out and helped peel the bark off of some ash trap trees that were set up in the spring just to look for emerald ash borer larvae. And so that photo there, that wasn't EAB, but that was a Lepidopteran borer. And then, of course, another thing I do, pests are always moving. There's always new pests coming up on the radar. So I do I attend conferences, do a lot of training. And so some of the photos on the right, we took there was a inspector's training over in Minnesota that I went to last fall. And so a bunch of different plant health specialists from different states kind of came in, and it's kind of another way to collaborate and kind of share information with each other. And we do these greenhouse trainings. So that was a photo of me, or I'm not in that photo, but that's a photo of us looking at a honey locust that was planted too deep and had some pest infestation. The Japanese apple rust, we saw that on our training, which was pretty cool because that was the first record of Japanese apple rust in Minnesota. So we came across that, and they hadn't found it in Minnesota until then. And then that photo below is just some borer that we found in one of the trees over in the trees. So yeah, another thing I do is outreach events. Like I mentioned before, outreach is super important, and I hope to do more of it this year. So just earlier this year, I've been to a couple of different conferences that were actually held here in Manhattan. So we have our booth set up. We went to the Turf and Landscape Conference hosted by the Kansas Turfgrass Foundation. There was the Shade Tree Conference hosted by the Kansas Arborist Association. And that's pretty much what our typical table looks like. We have, free samples. And we got handouts. We have insect specimens and collections. And then we have our board that has information on a lot of the pests that we look out for, just kind of letting people know, like, hey, look out for these pests. They're either here or they're going to be here, which kind of leads me to, talking about our insect watch list. So there's a lot of insects. We have a pretty decent sized list now of insects that we keep an eye out for. Some of them are here, some of them aren't here yet. The ones that we haven't detected for, but we like to talk to people a lot about are spotted lanternfly, obviously. That's probably going to be here eventually. So we want to, keep people aware of what to look out for in case it gets here. Asian longhorn beetle is another one. It's still a long ways off, but it can easily be brought in through human activity. And then another insect on our watch list is the walnut twig beetle. We actually survey for that. It's more native to the Arizona area, but we haven't found it detected yet. And then a few other species. So we talk about the spongy moth, red imported fire ants, Japanese maple scale. Those are all insects on our watch list that actually have been detected and or intercepted here in Kansas at one point. A few other species on the list. We got brown marmorated stink bug, emerald ash borer, of course, and spotted winged Drosophila. So those are just some of the species, we still talk about, in our outreach programs, but they are here in Kansas. All right, so that's a little bit about my position. Now I just kind of want to share with you guys what some of my plans are for the upcoming growing season. So like I mentioned before, spongy moth was and has been detected in Kansas in the past. Most recently, just last fall, we detected 2 male spongy moths in Johnson County. And they were found in traps that were set out by APHIS. And so this year, we're going to be doing a spongy moth delimiting survey. And, I haven't really ever done a delimiting survey before, so I volunteered to help with that. So I'll kind of be leading that survey. I think it involves setting out roughly 500 traps throughout the year. So that'll be a really good experience. Hopefully we don't find any more, but we want to make sure, yeah, we found these 2 male moths, but we want to make sure there aren't any more, populations out there. I know for one of the sites, it was found near a landscaping company, so it's probably likely just a hitchhiker. But yeah, that's why we do these surveys. And of course, every year we survey for emerald ash borer. Not too long ago, a few years ago, the state and federal quarantines were lifted on this insect. However, we still continue to monitor for it and do outreach in the hopes that, we can slow the spread and allow research and academia to have time to come up with solutions, either to control EAB better or protect our ash trees. So this year, we're going to be making trap trees in Southeast Kansas. And so what that involves is going down to the counties where you want to see if EAB is there. You girdle the bark and the phloem from the ash trees, which induces a stress response in the tree. It makes it really appealing to any emerald ash borer that are in the area. So if there is any there, they go to that tree. We leave that tree for the year, and then we come back in the fall and we peel the bark and we look for those signature S-shaped galleries that the emerald ash borer larvae make as they're tunneling. And if we find them, then we can say that, yeah, EAB is in this county. So we'll be doing that this year. We also do a walnut twig beetle survey every year. That's through our CAPS program. So we have a coordinator that kind of runs this survey and hires the staff for it. CAPS being, our cooperative agricultural pest survey. So we get funding for that every year. Walnut twig beetle is a vector for thousand cankers disease of walnut trees. So it carries that fungus into the tree, causing cankers. And, if the infestation is significant enough, it causes death of the walnut trees. And for us, these surveys are required every year because some countries that are receiving our wood products require that we survey for this insect. And so this year, last year we did most of the northeastern counties of Kansas. This year we're going down to the southeastern counties. That includes 67 trap sites, 19 counties total. My job is to support and train the temp staff. So we hire, I think, one staff member, and they go out and set out the traps. They check them, they bring back the traps, and then I pre-screen all those samples, look for any kinds of ambrosia beetle that resembles walnut twig beetle to the best of my ability. And then any kind of suspect specimens, I send them off to Carnegie Museum of Natural History, where there's someone there that can identify the beetle's two species. And so this was kind of one of the first things I did when I started my job in July last year. Just went through tons of different samples looking for walnut twig beetle. We're also doing another CAP survey. We're doing a vineyard pest survey. So we're looking for a few different pests of vineyards, and one of those being the Christmas berry webworm. We're also looking for the European grapevine moth, and our temp staff is going to be doing a visual survey for spotted lanternfly while they're out there. These vineyards are kind of all across the state. So that's 45 sites in 24 counties. And for me, I'll just be, again, pre-screening these samples for the two moth species. All right, and then finally, we're going to be doing a spotted lanternfly survey. This will be over on the far west side of the state in Colby, Kansas. We do this survey as kind of a follow-up from the dead specimen that was found in a 4-H collection in 2021 at the Kansas State Fair. It got a lot of media attention. However, for us, because the specimen was very desiccated and dried up and dead, we couldn't actually pinpoint its origin. So that's why we say, we haven't really found spotted lanternfly in Kansas yet. But because of this specimen at the state fair, the first year it was found, they did a thorough survey of the area. I actually helped with the survey last year. We went back to the town, looked at all the tree of heaven that was there. We looked for adult spotted lanternfly, nymphs, and honeydew, which would indicate signs of feeding. And we didn't find anything last year. And then this year, we're going to do one more third and final survey. And if we don't find anything this year, then that will complete our spotted lanternfly survey. And the picture on the right, we came across that last year on someone's property. It's actually a tree of heaven that was very well pruned and used as landscaping. So we thought that was pretty funny. But if you have tree of heaven, we recommend removing it since it's the primary host for spotted lanternfly. And then just a couple more things I wanted to bring to everyone's attention. We do have an insects webpage, and one of my responsibilities is keeping that webpage up to date. So there's information on current quarantines. Our insect watch list is on there with all the species, as well as a lot of information about them. We have we have last year's reports, we have previous year's reports on, survey results, pests to look out for, just a lot of good information that, we kind of encourage people to read up on, and so they can know kind of like what we're doing and what they can look out for. And then, of course, if you think you found an insect, please report it to us. If you see something suspicious, we recommend you take a photo and send any and all information to our email, kda.ppwc@kansas.gov. All those emails usually get forwarded to me, and then I'll get back to whoever and let them know if it's an important insect or not. Also, there is a public kind of reporting webpage that we use, and we encourage the public to report to. It's called edmaps or eddmaps.org. This is a great website where we can kind of collectively report our findings. They have professional verifiers that will say whether or not your record is the actual pest that you're reporting. So it's useful for us, and it's useful for the public if they're interested in knowing where pests are or have been found around the state. So just as an example, here's a map with all the points where tree of heaven has been found. And this might be useful in the future if spotted lanternfly is detected in some part of the state, and we want to look for all the tree of heaven and check for spotted lanternfly. We could go to this map and be like, hey, we know where tree of heaven is in this area, and then we can quickly save time and go to those sites. Okay, and so that's all I have. I wanted to thank you guys so much for listening and hosting me today. I hope you learned a lot, and I'm always looking for collaboration. So if you ever want to reach out with questions or need help with anything, please let me know. That's my contact information. And thank you all for listening.